Welcome everybody to this webinar on understanding your controls for Western blotting. My name is Will Olds. I'm the scientific officer for Protein Tech Group. I've been working on a lot of the technical content for the last 5 years, and I'm very excited today to tell you a bit more about how to use controls and which controls are best for your particular experiments. So in terms of our agenda today, what we're going to work on is why do you run controls in the first place, some considerations when choosing controls, going through a few popular controls, and then ending with protein ladders, one final control that you need in your experiments. So for those of you who are not familiar with Protein Tech, I just wanted to give a short intro here. So at Protein Tech, one of the major things that we do is all of our products are manufactured and validated in-house. So that gives us a lot of quality control as well as to be able to really know everything about the antibody that you're purchasing. And so we recently received ISO certification, which just means that we have a process in place for keeping things at a very high quality and reproducibility. And it also allows us to sell GMP grade products as well. We have 5 sites globally with full product inventory. Our products have been cited over 70,000 times in publications worldwide. And we also love to support the scientific community with mentorship awards, travel grants, workshops, and more to help you guys become the 70,000th and first publication. Our slogan is from our bench to your bench. And so what do we mean by that? So in terms of how the antibody industry is now, we have a bunch of different manufacturers, as denoted here, and they sell to resellers, and sometimes the resellers sell that stuff to you, or they sell it between themselves, or they sell it to other manufacturers. So what this means is that to you, the customer, you might be getting an antibody from A, B, C, and D, but they could all be coming from manufacturer A, for instance. But they're relabeled and said in such a way that you don't really understand, that you don't really know beforehand that you're bought 4 vials of the same exact thing. So at Protein Tech, we sell our stuff, only our stuff, and we sell it directly to you. And so you don't have to worry about this kind of a web of purchases in between these different vendors. It's going straight from, everything that we make is straight from ourselves, and it's completely unique. And so in addition, as I said before, that gives us kind of a little bit more lot-to-lot consistency, because reseller D may get it from manufacturer D now, but they may get it from manufacturer C later, and so the consistency may not be the same to you. And as well as being able to provide all our original validation data, including our raw films, and we have great stock inventory as a result of being able to control everything. So a little bit more about our products. We have antibodies against 13,000 human targets. We have 2,500 targets that have been validated by knockout or knockdown validation. We have more than 200 ELISA kits available. We also sell a line of humankind, human cell-expressed cytokines and growth factors for use in cell and gene therapies. Our Corelight brand of fluorescent dye conjugated antibodies, neutralizing antibodies, and pre-stained protein markers ladders. And so for us, you know, it's really about helping you guys. And so we really love to celebrate the success our scientists have had, and we're very proud of the great discoveries they've made. So below here, you can see some cover stories that have studies that have used our antibodies. And we've been cited in over 70,000 times in publications worldwide. And you can see kind of this graph that's kind of got a steep slope. So we're really excited to see that we're making a big impact in helping you guys make the next great discovery. Okay, so now let's get into the meat and potatoes of this. So why are controls and antibody applications so important? So first of all, it ensures that your results are biologically real. Otherwise, you may there may be artifacts, background signal that make it challenging to understand whether this is a real sample or not. And as well as minimize the variables that may be due to human error or environmental changes or what have you. It also allows you to compare data with your results, other lab members, as well as published data. So that's kind of more broader why controls are needed. let's go into Western blotting specifically. So let's say you did an awesome experiment here using HeLa cells, and for one, you're looking at this protein LDHB, and you're just validating and make sure it works. And so you have two sets of cells, one set of HeLa cells that you transfected with a siRNA scramble, denoted here, and then there's one where you used an siRNA directed towards LDHB. And so as expected, when you add the LDHB siRNA in there, it decreases the amount of signal in there, denoting that this antibody is specific. And so, you try to send this to a reviewer or something, they're not going to be very happy with you because you don't know why. One reason why this is low, why this is less intense, maybe because the siRNA worked, but it could be that your transfer was not complete at that angle for whatever reason for that lane. It may mean that you didn't load as much protein for whatever reason in that one. And then later, if you want to quantify, you can't really say if this one's really much lower than this one because there's no benchmark that's used. And so that's why you need something like tubulin in this case. And so as you can see here, this tubulin is the same between both lanes. And so that means the same protein was loaded, the transfer was right, the blocking, everything worked out just as you expected. And so therefore, if you compare the signal compared to tubulin here, and then compared to tubulin here, you can see that there was a significant drop off here. And so therefore, you can say that the siRNA had an effect on this target. So what makes a good control? So a great control is a ruler, a benchmark, something that should not change despite the other variables in your experiment changing. It needs to be something that's ubiquitous, something that you can use throughout your experiments and not have to change or swap out things that frequently. It needs to be something that's highly expressed, and it needs to be an essential gene that's not affected by many cellular changes, as well as being, and so for the most part, these are structural and metabolic proteins. On the right here, we have a Western blotted GAPDH, which is one of the favorite proteins that people use for their loading controls. And this one's really excellent in that it can be used for all the way from zebrafish to HeLa and heart. And I think some people have even used this in yeast as well. So it's a really wonderful control. So when choosing a control, you don't just kind of pick from a bag and say, I'm going to pick GAPDH today or beta-actin this time. You need to ask yourself a couple of questions. So the first one is just like, does the experiment affect the control in any way? If it's changing GAPDH levels, then GAPDH is not going to be a great control for you. It's also important to check out the literature for your specific field. There could be controls that people like to use for that particular thing, and it's best to follow the convention of your colleagues in that field. Additionally, if you're analyzing organelle fractions, it's important to choose one that's specific for that organelle. So again, if you use like GAPDH, which is in the cytosol, and you're looking at a nuclear fraction, you're not going to get any signal, and it will not be a good control. The last consideration is how close in molecular weight is my target protein to the protein that's being, is the control to my favorite protein that's being analyzed. So for instance, GAPDH is 37K, and so a 30 to 40 kDa protein is not a good choice. And also, again, like think about like beta-actin as circadian variation. So for certain experiments where you're looking at circadian rhythms, it's not the best control to use. So here's a handy chart for all of your loading controls. And so it's listed here from high molecular weight to low molecular weight. So you have kind of the all-stars that we all know and love here. But if you're looking at a high molecular weight protein, vinculin's really great for the whole cell. And then if you're looking at nuclear proteins, here are some great ones like lamin, HJAK1, histone H3. And then for mitochondria, there are also some other ones there. COX-4 for a little bit lower, VDAC for more your kind of mid-range, and for serum samples, transferrin is a really great loading control. So just kind of want to, so I'm going to highlight a couple of these products here that we provide. So first one I want to highlight here is beta-actin. So this is a mouse monoclonal antibody. And on the left here is serial dilution using the same samples and with different dilutions, progressing dilutions of antibodies. So for this first one, this is using this product here from 1 to 40,000 and all the way down to signal that you can see at 1 to 64,000. We then compared this to, on the same experiments, we were also comparing this to ones from other companies that provide this. And as you can see here, the signal drops out precipitously as you do this. And the Protein Tech one is quite superior. And it has, so its max dilution is about 1 to 640,000. So it's almost like one vial is a lifetime supply. And for the competitors, it seems like for this one's 1 to 320,000 and this one's 1 to 80,000. But the main thing I kind of want to keep back here is that, you know, you're getting a lot of bang for your buck here. The cost is much lower here so it's about $99 in the US and about 150 euros at this moment. You can see how this is much cheaper than the competitors. Over here is just an siRNA experiment showing that this is a specific antibody towards this target. So the next one I want to highlight is GAPDH. And so here GAPDH is we did a very similar experiment with serial dilution and just looked at the see when kind of the signal goes out. And so we this one, you know, we were able to go up to 1.1 to 1.2 million. And it looks like one of our competitors was also able to go to that same level. However, this other one did not perform quite as well. And so again, this one has similar performance between these two, but this one's again much cheaper. So this is a real bargain for what you get out of it. And on the right here is the same Western blot I was showing before, that for this particular antibody, showing that it just works across a variety of cell lines and tissue types. The last one I just want to highlight here is vinculin. So this is about 110 kD-ish, and this one is really excellent for high molecular weight markers and works across a variety of cell lines, as you can see here. So when you go to any website looking for these targets, you're going to see a monoclonal and polyclonal options. And so you might wonder, which one should I choose? So monoclonals have high lot-to-lot consistency. However, because they have a single epitope, they may have limited species reactivity. So this is always something to check. Polyclonals have a bit lower lot-to-lot consistency, but have a greater chance of reactivity to non-mammalian organisms such as yeast and Drosophila. So then the next thing to kind of look for is pre-conjugated primary antibodies. And so these you sometimes see in the market. They'll be HRP conjugated to GAPDH or beta-actin, or a fluor or something else that you could use for a Lycor machine, for instance. And so when should you use these? When should you not use these? So in terms of pre-conjugated primary antibodies, the advantages are that you don't need to worry about the secondary antibody for your control. You don't have to worry about thinking about, okay, I need to add this secondary and it'll work. You have one that's already there. So really, it's a lot of time and resource saving for you. The disadvantages is that whatever one you pick, it's really compatible with only one type of system. So you just need to make sure that whatever you buy is compatible with your imager that you're using for your Western blots. Another one is that because these will generally only have 1 or 2 of these molecules on their conjugates, that for single development, the sensitivity may be a little bit lower than a traditional secondary that's where you're able to get a bunch of molecules onto a single IgG to amplify the signal. The last disadvantage is that it tends to have a shorter shelf life. Anytime you conjugate an antibody with something, the stability of that conjugate is quite low. However, having said that, these are a great way to, a lot of customers really love these because you're able to save so much time and resources for this. So again, I just want to highlight kind of the importance of positive and negative controls. And so for positive controls, we recommend really any cell type or tissue with endogenous protein expression. Great resources for finding out good positive controls are just what are one, whatever cell types or tissues the antibody manufacturer uses. And then the second one is online resources such as PaxDB and uniprot.org. Both of those are able to tell you which cells or tissue types express this protein. Negative controls can be a little bit more challenging. Most cells express a lot of proteins at low levels, and so it's very challenging sometimes to find a cell type or tissue not to express the protein using those resources I mentioned before. So the best kind of one to use is one where it's been genetically knocked down or knocked out with CRISPR or siRNA. So here's like but on the right here, I show an example here where for this particular target, you take the mouse spleen does not express it, so it's a great negative control for this particular target. And so, as I said before, a major part of this highlight of these antibodies is their price within the market. So in the US, 99, and it's less than 150 euros. And this is for GAPDH, for instance. We have customers who really love this antibody, as you can see here. And so here are just a couple more of our popular loading control products that you can see here. I'll allow you to get a second so you can just write them down if you'd like. And lastly, you don't want to forget a molecular weight ladder. And so we sell a couple of these. So the first one's kind of our standard one that's kind of a bread and butter that'll work for a lot of different things. And so for this pre-stained protein markers, it has a molecular weight range from 10 to 180 kD. And we have 33 customer reviews, all are 5 stars. And people just are really loving this. And so an example blot is on here. So as you can see, the colors are very easy to distinguish, and a couple colors in there so that you can see the different molecular weights. So that's for kind of the, as I said, that last one's kind of the standard one that people use. This is if you're looking at a broad range. This one goes from 3 to 245. So enormous proteins can be used. This is best for huge proteins or really, really small proteins. And this one's also relatively highly reviewed as well. So our take-home message today is that controls are vitally important in antibody applications to ensure the validity of results. And as always, read. Research which controls are best for your experiment and your application. Always run controls alongside your experiment. And be critical of your antibody supplier, even if they are protein tech. Ensure that they have properly validated the antibody and can show the original data. So here are some of the useful resources I mentioned before: uniprot.org, which has protein sequences, variations. It tells you a lot about post-translational modifications, and everything's been is linked to a publication or another resource that you can trust. PaxDB is a great, as I said before, is great for tissue expression and comes from paper. All the information comes from papers as well. And then our website also has application-specific tips and tricks and product-specific protocols. So with that, I want to say thank you for listening, and feel free to email me at will@ptglab.com if you have any questions. Thank you for listening.